Hey guys, we're going to be talking today about protein synthesis. This is your third lecture. Make sure you have in front of you a pen or a pencil, marker, crayon, paintbrush, piece of chalk, I don't care, just as long as you got something to write with, and a piece of paper to write your notes down on. Okay? The title of this lecture is Protein Synthesis. Our objective today: at the end of this lecture, students will be able to analyze the differences between types of RNA in order to identify their separate roles in protein synthesis. Okay, make sure you have that down in your notes, right underneath your title, so you can keep yourself organized with each of these lectures. We're going to start out by comparing DNA versus RNA. So we've talked about DNA already. We know the sugar type is deoxyribose. Remember that ose is a carbohydrate, it's a sugar. The structure is a double helix, it's double-stranded, it's like that ladder, that twisted ladder. And the bases are A, T, C, and G. So we've covered that pretty thoroughly in class, and you've already taken a quiz on it. And we're moving on to RNA, which is a little bit different than DNA. Okay? So RNA, the sugar, is ribose. Notice that in DNA, deoxyribose begins with a D, and ribose begins with an R, so that can help you keep track of which one's which, DNA and RNA. Okay, the structure of RNA is single-stranded. It is only half of that ladder. And the bases are going to be a little bit different. Instead of T, we're going to have a U. So we'll still have A, C, and G, but we're going to replace that T with a U, and I've made it giant and bold for you. U stands for uracil, and A will always bond with uracil. So, to get that down in huge block letters in your notes, highlight it, star it, circle it, underline it. Adenine bonds to uracil, only in RNA, excuse me. Okay, so A and U go together. Cytosine will still bond to guanine in RNA. So C still bonds to G. All right. So what is protein synthesis? It is the process of using DNA code to make proteins. Proteins, as we know, are found in meats, cheeses, eggs, milk, types of things like that. Proteins are everywhere. They make up your entire body, and they determine your traits, like your eye color, your hair color, the length of your fingers, how your fingernails grow, how your teeth are going to look, all that stuff. There are two major steps to protein synthesis, and that's transcription, which takes place in the nucleus of your cells using mRNA, and translation, right here, which takes place in the ribosomes using tRNA. And remember, ribosomes, we already know that's where proteins are made. So now we're going to be learning more about that. All right. So I have a short cartoon for you to watch on protein synthesis. All right? And we're going to see firsthand the process of using DNA code to make proteins. When watching the cartoon, I have a few look-fors for you. First one is look out for mRNA. We're going to see where it exists and what it does. Look out for tRNA. And lastly, look out for rRNA. Okay? All these things are going to be in the video. So, we'll get started on the video and we'll come back to this when it's done. All right? Let's see.
This is a cell. Cells are capable of metabolizing their own nutrients and building proteins. Each cell has many parts, such as a membrane that helps move things in and out of the cell, and a nucleus. A cell's nucleus controls the activities of the cell and houses its genetic material, or DNA. DNA consists of units called nucleotides, which are made up of a sugar and phosphate backbone, and one of 4 molecules called nucleobases. You could describe a strand of DNA as looking like a twisted ladder, with the nucleobases connecting it in the middle to make up the rungs. These nucleobases are adenine, thymine, cytosine, and guanine. They join via hydrogen bonds so that they are complementary to each other. An adenine will only bond to a thymine, and a cytosine to a guanine. During protein synthesis, a strand of DNA needs to be copied and taken out of the nucleus. This process of copying DNA is called transcription. Transcription starts with several enzymatic reactions that unzip a strand of DNA. The unzipped strand is used as a template for synthesizing a complementary strand of messenger RNA, or mRNA. The mRNA is composed of nucleotides similar to that of regular DNA. The only difference is that mRNA has uracil in the place of thymine. Once the DNA has been copied or transcribed, the mRNA leaves the nucleus and enters the cytosol. Here also are the amino acids for protein synthesis. Some of these amino acids are from proteins previously digested, absorbed from the gastrointestinal tract, and transported to cells. Ribosomal RNA, or rRNA, binds to the mRNA and aligns it to begin a process called translation. During translation, the rRNA moves along the mRNA in segments of 3 nucleotides at a time. These segments are called codons. Each codon specifies for a particular amino acid, which is carried over by a specific transfer RNA or tRNA. Any tRNA that has its designated amino acid bound to it is called an aminoacyl tRNA. The tRNA also houses an anticodon, which aligns to the codons on the mRNA. By recognizing the codon as well as the specific amino acid, the tRNA translates the codons into the amino acid sequence of a protein. Each translation is initiated by the codon for methionine. When the aminoacyl tRNA is in place, the ribosome will move to the next codon, and another aminoacyl tRNA will arrive with the next amino acid. As each new aminoacyl tRNA gets in place, a peptide bond is formed between the new amino acid and the peptide chain associated with the previous tRNA. The growing peptide chain is now bound to the new tRNA, and the previous tRNA breaks off and leaves. The chain of amino acids keeps growing longer and longer. Eventually, a stop codon is reached that is not recognized by any tRNAs. This is the signal that tells the process to stop. The newly made polypeptide chain is then processed to form a new protein.